Here we present a study using focused ultrasound-mediated disruption of the blood-brain barrier in order to improve the delivery of a gene therapy targeted to the brain of a mouse model of Huntington's disease. Huntington's disease is a hereditary neurodegenerative disorder caused by a CAG repeat expansion in exon 1 of the HTT gene. The presence of this mutated protein leads to cellular toxicity and ultimately neuronal cell death. Several gene therapy strategies aimed at treating this disease at its source by lowering the expression of the mutated HTT protein have shown promise, with several reaching clinical trials. However, delivery to the brain across the blood-brain barrier remains a major hurdle to success. In our study, we are targeting FUS-BBB opening to the right striatum of both wild-type and HD model mice. The aim of the first study was to determine the best combination of FUS pressure and microbubble dose to get the best delivery across the BBB. This was performed in wild-type mice using an AAV1 capsid that is known to normally not cross the BBB. The aim of the second study was to validate the approach in the HD mouse model using a more clinically relevant AAV9 capsid, also with a GFP tag, and this was done at the optimal parameters that were determined by study 1. These are the results from study 1, where the top row shows the contrast MRI data acquired after BBB opening. And as expected, the disruption increases with increasing FUS pressure and increasing microbubble dose. The bottom row shows immunofluorescent images of the GFP signal, but here the same pattern is not followed. And in fact, the parameters that were chosen were the lower FUS pressure along with the higher microbubble dose. This slide shows a close-up of the two mice that were done at these parameters, showing good delivery of the AAV1 vector to both the targeted side, and we also got some delivery to the non-targeted contralateral side. And this is in contrast to the control groups where no focused ultrasound was applied, with the AAV1 delivered at the same dose level, where you can see there was essentially none that got across the BBB. This slide shows an example of the kind of data that we acquired in study 2, where we were delivering an AAV9 GFP capsid to the YAC128 mouse model of Huntington's disease. The top row is showing an example from a single mouse of an immunohistochemistry stain slide for GFP signal, and we also acquired markers for microglia activation for an IBA1 stain and astrocyte activation with a GFAP stain. In order to pool the data over all mice in study 2, the contrast MRI data was co-registered to a mouse brain template, and then ROIs were extracted for the contrast enhancement in both the left and right striatum. Similarly, for the immunohistochemistry data of the GFP signal, several slices were used to cover the full striatum, and hand-drawn ROIs were used to define both the left and right striatum. Here are the individual mice that we looked at, showing the contrast MRI data in the first column and the immunohistochemistry stain GFP signal in the second column. And then the bar plots are showing comparisons of the targeted right striatum against the non-targeted left striatum, and looking at the data in 3 different ways. First, looking at the global mean signal of all the pixels in those ROIs, where you can see that did not show up as a significant difference for the GFP signal. And then looking at the mean of the top 10% of pixels within those ROIs, which did just reach significance, or considering the percentage of the pixels in the individual ROIs that were above a certain threshold, which did meet a significance criteria. So in conclusion of this very preliminary study of doing delivery of AAV capsids to mice, we've shown that both an AAV1 and an AAV9 capsid can be delivered across the BBB in both a wild-type model and a model of Huntington's disease. We did some initial optimization work on the different parameters, but there's likely more work to do and more parameters we can consider. While we've shown delivery, the next big question is, does this amount of therapy lead to a therapeutic effect? So our next plan for the future will be to do a therapeutic efficacy study in which we deliver an AAV9 microRNA targeted to lowering the mutant Huntington protein expression.